Thanks for coming back from the coffee break, hopefully refreshed to listen to the next 4 case studies. We start with Rosalind Leggett. She is a biologist and scientific advisor for fisheries and oceans in Canada, and she examines the potential of environmental consequences of GM finfish. And her talk is about problem formulation for the environmental risk assessment of GM growth enhanced salmon. Rosalind, please.
Very much, and thank you for the invite. Okay, so I'm going to discuss the problem formulation for environmental risk assessment of genetically engineered fish. And in this case, I'm going to use a test case of growth hormone transgenic coho salmon. So in Canada, regulation of genetically engineered fish is under the Canadian Environmental Protection Act, or CEPA, New Substance Notification Regulations for Organisms. So if a genetically engineered fish is notified under CEPA for proposed commercial use, a risk assessment is performed by Fisheries and Oceans Canada, my department, using the classic paradigm. Risk is proportional to the exposure and the hazard, and we include a measure of uncertainty in both the exposure and the hazard. This risk assessment is peer-reviewed and published in the public domain, and it provides the basis for science advice to the departments that are tasked with the regulatory decision on the use of genetically engineered fish under CEPA. And that's Environment and Climate Change Canada and Health Canada. So risk assessment generally begins with problem formulation. In our case, we define the scope of the risk assessment as well as our protection goals. We provide a description of procedures for concluding on risk, as well as a characterization of the organism, the comparator species, and the ecological parameters of potential receiving environments. So there's a couple of genetically engineered fish that have been approved for commercial use in Canada. The most well-known is the Aqua Advantage salmon. This is an Atlantic salmon with a growth hormone transgene, which is used for faster growth for land-based aquaculture, and this is currently under assessment for expansion. A little less well-known is the glowfish electric green tetra. This is a little white tetra that contains a cnidarian green fluorescent protein transgene to produce a new color morph for the ornamental aquarium trade. So you can now go to the pet store and buy your own transgenic fish. And there are currently 5 other transgenic tetra lines for under assessment under CEPA. So I'm going to use growth hormone transgenic coho salmon as a test case for environmental risk assessment. Now this is a research line of transgenic fish containing a sockeye salmon growth hormone transgene driven by a metallothionein promoter, and this results in fast growth. So this is a research line that was developed by Dr. Robert Devlin in the 1990s at Fisheries and Oceans Canada, and its purpose is to examine the impacts of accelerated growth in salmonids, so it's not actually for commercial use. But it does make an interesting test case because its physiology and ecological impacts are very well studied. So the theoretical use scenario I'll discuss is land-based aquaculture of triploid, sterile, all-female growth hormone transgenic coho salmon in a land-based facility at the current site of our research facility, which is in BC, Canada, and it's within the natural marine and freshwater migration ranges of wild stocks of coho salmon. So for simplicity's sake, I'm going to discuss risk protection goals focusing on the impacts through hybridization with wild salmon, although under CEPA we would look at a lot of other pathways to harm. So what are we trying to protect? In this case, it's the maintenance of wild coho salmon stocks. Coho salmon are anadromous fish, so they spawn in rivers, and the juveniles live in rivers and lakes for about 1.5 years, at which point they migrate to the Pacific Ocean and undergo huge marine migrations before returning to their home rivers to spawn at 3 years of age, and they die after spawning. So this homing to specific rivers to spawn results in many genetically and ecologically distinct populations. They have ecological importance in both marine and freshwater food webs as both predator and prey. And the mass die-off at spawning, wrong one, sorry, results in a huge influx of marine-derived nutrients into riparian ecosystems. So this has a huge import in nutrient cycling as well as biodiversity in these ecosystems. They are very important in both commercial and recreational fisheries, as well as have cultural importance to local indigenous communities. So they are both very socioeconomically important as well as ecologically important. So a lot of interest in protecting them. Like many lucrative fishery stocks, they are currently at historic levels, and they are threatened by overfishing, climate change, habitat destruction, among other things. So the question is, could this theoretical land-based production of growth hormone transgenic coho salmon further threaten wild coho populations? So what are the growth hormone transgenic salmon? So the targeted phenotype is this greatly accelerated growth compared to wild-type fish, as well as improved feed conversion efficiency. So these are two very important traits for aquaculture. However, the insertion of the growth hormone transgene results in a number of off-target, unanticipated effects. And these include pretty much every major life history characteristic that's been examined has been affected. So in terms of life history timing, the fish mature at a usual size, but at 2 years of age as opposed to 3 years of age. They have great differences in gene expression, disease resistance. Behavior is an interesting one. Armando, can you get the video going? All right, so this video is feeding time with the wild-type fish on the left and the genetically engineered fish on the right. And you can see that the wild-type fish are balancing predator avoidance behavior with foraging behavior. But the transgenic fish are basically, their accelerated growth is motivating them to feed. So they're always hungry, and it's basically overriding any predator avoidance behavior they have. So the potential effects from both the targeted as well as the off-target effects must be considered when conducting an environmental risk assessment. Another important thing to consider is genotype by environment interactions. So this is when one or two or more genotypes respond to different environments in different ways. So the best way to figure out if transgenic fish could cause harm to a natural ecosystem is to put them in a natural ecosystem and see what the effects are. We can't do that for many, hopefully obvious reasons, so we have to try and predict what would happen in nature using laboratory studies. However, we found that the transgenic and wild-type coho salmon respond to different environments in very different ways. So this is an example in freshwater, where in the hatchery situation, in a simple environment fed commercial diets to satiation, the transgenic fish get this accelerated growth, advanced life stage, compared to wild-type fish. But if you rear these same fish in a simulated stream tank, fed natural food items, the wild-type fish are not that much different, but the transgenic fish now actually look an awful lot like a wild-type fish. So you can imagine the potential risk effects of these two fish might be very different because of their phenotype is so different. This is also seen in the marine environment. In this case, when we rear wild-type fish in small-scale laboratory conditions, they have an extremely stunted phenotype compared to what the same fish would look like if they were reared in nature. And this stunted phenotype isn't really seen in the transgenic fish under small-scale culture. When we rear the fish in seawater mesocosms to try and decrease these effects of culture, again, we see two different phenotypes. So there's strong genotype by environment interactions here, which make predictions of success in nature using laboratory-derived data quite difficult to do with acceptable levels of certainty. And this also identifies the importance of not relying solely on data from simplified culture environments when looking at risk. So the pathway to harm is a good kind of exercise in problem formulation to see what steps would be needed to be completed for the transgenic fish to harm wild populations. So I'm going to go through an example scenario, pathway to harm through hybridization with wild populations. So in this case, it would require the release of the coho salmon, survival to maturation, migration to spawning grounds at the same time as the wild-type fish, successful mating with the wild fish, and then the F1 hybrids resulting in decreased wild coho productivity, either through poor survival of the hybrids or if introduction of the transgene had resulted in deleterious alterations in the genetic structure of the wild populations. So I'm going to go through what scientific research might be needed or is available to address each of these steps. So for simplicity's sake, let's say the fish have escaped. So the next step would be survival to maturation. Now, survival in a culture environment for most genotypes is really very high and is not a good proxy for what you might see in nature. And we found studies in semi-natural conditions, the relative survival of the transgenic fish varies greatly compared to the wild-type fish, depending on the time of the sort of release of the fish to the semi-natural conditions, the disease status of the released fish, the availability of food, the presence of predators, things like that. So just to give you two examples, and we have many of these. So this is in a simulated stream tank from first feeding until smolt. And we see under one food level, the survival of the transgenic fish is very similar to the wild fish. But then on a different food level, the transgenic survival is actually lower. In the marine environment, from smolt to maturity, what we see is that when the fish are from a population known to harbor a chronic bacterial infection, the transgenic survival is much lower than the wild-type fish. But if the fish are taken from a population known to be negative for that chronic disease, then the survival of the two groups is very similar. So data so far shows that survival is possible, although the relative survival to wild fish is expected to vary depending on the conditions that the transgenic fish might encounter. Next is migration to spawning grounds. This has not actually been examined in growth hormone transgenic coho salmon. The main commercial product would be triploid sterile females, and in salmonids these fish do not mature, and it's been shown in other salmonid models that they also don't migrate to freshwater spawning grounds. So that particular group is not expected to migrate. However, there would be, triploidy is not 100% effective, so there'll be diploid exceptions in there, as well as diploid fertile broodstock. So domesticated Atlantic salmon can be used as a surrogate as a fast-growing model for these fish. And it's been found in domesticated Atlantic salmon that while many farmed fish do not actually make it to the wild spawning grounds, there are recurring instances where they do. The location of the proposed aquaculture site is within the migration range of many wild coho populations, so there's no physical impediment to them getting to these wild spawning grounds. So we can't exclude the potential for diploid escaped transgenic salmon to migrate to spawning grounds. So migration is predicted, but it's not empirically tested. Next is successful mating with wild populations. So in early studies, we found that laboratory-reared transgenic fish had extremely poor spawning success when spawning in competition with wild-reared, wild-type fish. However, the wild-type fish reared in these same conditions also had extremely poor spawning success. So there was a huge effect of culture. When we reared the fish in mesocosms to try and minimize these effects of culture, spawning success was improved, and in these conditions we found transgenic and wild-type fish had similar spawning success, although still much lower than the fish reared in nature. So successful mating with wild fish is predicted, although the level may depend on the time of release, in other words, the effect of culture, as well as the level of mate competition. And finally, successful mating with wild populations would have to cause a decrease in the wild productivity. And this can go through two pathways, through poor survival of the F1 hybrids, or if introduction of the transgene results in deleterious alterations in the genetic structure of the wild populations. So in terms of survival of hybrids, semi-natural stream conditions have found the transgene may decrease survival in some conditions, although in other conditions there may be no effect, and in different conditions there may actually be an increase in survival. And this depends on a number of factors, including the availability of prey, presence of predators, and the level of competition. In terms of the alteration to the genetic structure of the wild populations, computer modeling of the transgene integration found the growth hormone transgene in a wild population can potentially shift genetic backgrounds in both the transgenic individuals as well as the non-transgenic individuals, which could direct phenotypes away from the naturally selected optima. A recent study found quantitative trait loci mapping demonstrated the growth hormone transgene alters the genetic basis of growth-related traits in coho salmon, and the authors postulated that this might influence evolutionary changes within the coho populations with ecological consequences. So current data suggests that poor survival of hybrids may be present in some conditions, and alterations in genetic structures with deleterious consequences of the wild populations could be predicted. So overall, all steps post-release have been corroborated to some extent in the laboratory or through use of surrogate organisms. So the potential harm to wild populations through hybridization with growth hormone transgenic coho salmon is predicted should these genetically engineered fish escape. However, the predicted level of success of the transgenic fish or harm from the transgenic fish differs when examined under laboratory versus semi-natural conditions. So this highlights the importance of simulating relevant natural conditions when examining environmental risk in the laboratory. Genotype by environmental interactions, as well as limited studies in some steps, result in moderate uncertainty in predicting the occurrence or level of harm in nature. And I should also point out that I've just looked at one simplified pathway to harm, but nature is never simple, and there are a number of different interacting pathways to harm that could occur. So because harm is predicted, the only way to mitigate risk would be to basically mitigate exposure. So this highlights the importance of containment to prevent harm to environmental components from the use of genetically engineered transgenic coho salmon. Thank you very much for your time.
Thanks very much, Rosalind, for sharing the outcome of the case study on the ERA consideration for growth hormone transgenic coho salmon and the importance of simulating natural conditions. And with this, I would like to open the floor to the audience for possible questions. Are there any questions for Rosalind? Yes, please. Can you indicate your name and affiliation, please?
Jeremy Sweet, environmental consultant from UK. Your final slide there, you suggested that physical containment was the only way of eliminating the risk. Mm-hmm. But what about biological containment? In other words, sterility systems?
So the triploid salmon, in theory, would produce a biological containment. Triploid is not 100% effective. It's quite high, it's about 98%, so it depends on the level of release that you would get. And triploid salmon could still cause effects, it would just be in one generation. Another thing is that you still need to have diploid broodstock, so they also have to be considered in the risk. But yeah, having multiple levels of containment, so physical as well as biological, would greatly decrease your chance of exposure.
Any other questions for Rosalind? I see none. Do we have any from the... here, sorry, please.
Yeah, Peter Dom, just a very short one. So what is the consequence? Do we allow this or not?
Well, in Canada, it is actually currently allowed. So it's, but it's a very, at the moment, the land-based facility, for at least for Atlantic salmon, is, it's many levels of containment. So their grow out is triploid all-female fish, and they're currently grown on a mountaintop in Panama. So even if they escaped, by the time they got to the water, it's too hot, and they would die. And even if they didn't, there's only females there, and there's no one to spawn with. So there's many levels of containment. They have applied for a facility in PEI in Rollo Bay for land-based aquaculture, but that's currently under assessment, so we'll see. I'm not going to comment on that yet.
So in Chile, you recently had a large escape of, I think, trout or salmon, I'm not quite sure. Can you use this as a case study for your situation in Canada?
Possibly. I mean, Chile is a little different in that there are, so if Atlantic salmon escape, there's no wild Atlantic salmon population to impact. So the biggest potential impact in this case is with the wild counterpart. So, I mean, looking at Canada has kind of two opposite coasts where on the Pacific, the wild stocks, although they're doing poorly, are still, they're measuring in the hundreds of thousands to millions, while on the East Coast, the Atlantic salmon wild stocks are, they're sometimes measuring in the dozens. So an escape of 1,000 fish there is different than escape of 1,000 fish on the Pacific region. So there's a lot of different factors that have to be considered. But basically, any information that we can get adds to what we know. So yes, it would be considered.
Thanks, Rosalind. Just to double check, are there any questions from the app? No? Okay, if I do not see any further questions, so thanks very much, Rosalind, again. Our next speaker is Andrew Roberts. He works at ILSI Research Foundation as a Deputy Director of the Center for Environmental Risk Assessment. Andrew works includes, next to many other, trainings and capacity building related to problem formulation for environmental risk assessment. And today he will present us a case study for the problem formulation for environmental risk assessment of gene-derived modified mosquitoes. Thank you, Andrew.
Thank you very much. And thank you for the organizers for inviting me to present here. I've really enjoyed the session so far today. I think I will refrain from making all the comments that are swimming through my head on all the interesting intellectual ideas that have been discussed today. So I'm going to talk about problem formulation for the environmental risk assessment of gene-derived modified mosquitoes. And the reason this is under consideration is because it's a strategy to eliminate malaria. And I think, one of the thoughts that does occur to me is that we do risk assessment to inform decision making. And I think as we strive for continuous improvement and to better inform those risk assessments, it's always important to keep in mind that the context is to help support decision making. And some decisions have very serious ramifications. Even if I stay on time, which I'm very much hoping to do, while I give this presentation, 11 children in Africa will die from malaria. And that's a conservative estimate. So people are trying to develop and deploy new technologies, not because they're technology enthusiasts. I have not met anybody who's just excited at the prospect of gene drives. They're excited at the prospect of trying to prevent death from malaria. So, this is why we undertake the task. And we were approached in 2006 by the Foundation for the National Institutes of Health because they were looking for somebody to help them organize an activity to address environmental and ecological risk assessment. And they didn't use the term problem formulation, but that's what they were looking for. And I've been fortunate enough to be able to, over the last 2 years, participate in these activities. This is about my organization, which is the ILSI Research Foundation. I'm going to go with the assumption that you'd rather hear about problem formulation for gene-derived mosquitoes. So this is here to go into the public record, but I won't talk about it. I love my organization. Please feel free to come and ask me about it afterwards. This is what I'm really going to talk about. I'm going to introduce the concept of gene drives. This is something that has been in the popular press, and I think it's important to understand what they really are. Then I'm going to give a very brief description of what we've done in the course of these activities. And then I'm going to go much too quickly through far too many slides that describe the results of those activities. And I hope you'll forgive me because I know that too many slides delivered too quickly is one of the cardinal sins of presentation. But I couldn't bring myself to cut any more, so I'll do the best I can. So what is a gene drive? In order to understand this, first you have to understand what happens to a gene when you introduce it into a population. And this is a slide showing normal inheritance. And it's worth mentioning that this is any gene introduced into a population, whether that's a transgene or a wild-type gene. If it's introduced into a population without any significant selection pressure, normal inheritance does not increase the presence of that gene in the population. So if you look, the red mosquito in the diagram on the left is carrying the trait. It mates with a wild-type mosquito. It passes on the trait to half of its offspring, as does each subsequent mosquito that's inherited the trait. And what you see after a few generations is the trait is still there, but it hasn't increased in abundance in the population. A gene drive is anything that changes that pattern, where you get non-Mendelian inheritance of a gene. This diagram uses a Cas9 mediated gene drive construct, but it's important to understand that gene drives exist in nature, and there are multiple mechanisms known from the environment that increase inheritance of particular traits, and they all work this way. So the parent has a greater than 50% chance of passing on the gene to its offspring, and it's easiest to illustrate if you use 100%, but there are gene drives that function at 60%, 80%, 95% transmission rate. So instead of passing on the trait to one of its offspring, the trait is passed on to all of the offspring of the parent that contains the trait. And then you can see in this simple diagram that very quickly, at least in theory, not in practice, but in theory, you can spread the trait through the entire population. And the characteristic of this is that it doesn't depend on any sort of selection pressure. And in fact, modeling suggests that you can drive a gene through a population using genetic mechanisms against a gradient of selection pressure. So even if the trait is not desirable from a fitness point of view, you can still spread it into the population. So this is what a gene drive does. It's a mechanism for spreading a trait through a population. And the idea behind this concept is a genetic biocontrol concept. And I think that phraseology is important, and I'm very pleased we'll have a biocontrol presentation next, because that's really what the proposal is. This is the introduction of an organism to control another organism in the environment. And in this case, they're of the same species. So just very quickly, this is a lovely slide prepared by my colleague. I'm not going to read all the words on here, but the idea is you can release a relatively small number, not one mosquito, but a relatively small number of mosquitoes in a relatively short amount of time without repeating this over and over again. And the gene drive mechanism does the job of spreading the trait through the population rather than you releasing the organism all over the place and actively maintaining it. So that once it's introduced, it takes care of the spreading. So it's an exposure mechanism. And the topic of our work has been to look at the use of gene drives in Anopheles gambii for the purpose of controlling malaria. So what have we done? Well, we've done 5 different workshops and consultations. The first one took place in Reston, Virginia, which nobody's ever heard of, but it happens to be right next to the Dulles Airport in Washington, hence the location. We did this in 2016, and the participants were largely subject matter experts on mosquito biology, ecology, environmental and ecological risk assessment, as well as a large international contingent from Asia, and especially from Africa, where there were African regional organizations and individual government risk assessors who came for context to be able to participate and say, well, you know, these are being deployed in Africa. What are our environmental protection goals in Africa that need to be considered? Coming out of that meeting, one of the participating organizations is something called the New Partnership for African Development, which I don't have time to describe to you in detail, but it is a development organization. It's part of the African Union, and that group in particular was very excited about the results of the workshop in Reston and insisted that we should do this activity, this same activity, regionally in Africa in order to build awareness, solicit input from African scientists and African regulators. And I think everybody liked that idea, including and especially me. And NEPAD proceeded to organize the next 4 consultations in Africa, and they invited us to facilitate the problem formulation exercise. But it was their workshop. We were just providing assistance. And those took place in Accra, in Nairobi, in Gaborone, and in Libreville over the next year and a half. The purpose of all of these activities, and I need to stress this, is to begin the conversation about how you will do risk assessment if you seek to use a gene drive for the purpose of controlling malaria. Nobody at any phase of these discussions thinks that the conclusions are the end of the discussion. There's recognition that it will take many more years to simply develop the technology, but the hope is to begin having these conversations so that people can better understand how to do risk assessment when the technology is available to use, because there is a compelling public health need for this and other technologies to control malaria. And then also to identify areas where researchers and risk assessors should be putting their effort into developing information that will inform risk assessments that may be done in the future, because nobody wants to waste time doing a lot of unnecessary experiments and collecting a lot of data that's uninformative to risk assessment. So to provide a rational starting point for these discussions. All of these activities were 3-day workshops. Normally I have a dozen slides explaining in great detail what we did, but I think because we've had such a great background in problem formulation today, we spent 3 days. The first day was an information dump. Here is the context that you need to understand if you're going to go through a problem formulation exercise. What is this mosquito? What is malaria? How does that work? What is the lifestyle of the mosquito? And I don't have time to go through the biology of mosquitoes. I'm hoping people understand what this organism is and how it works. Day 2, we used hypothetical case studies, so not real examples of work that's underway to develop gene drives, but realistic hypotheticals of how a gene drive might be used in Anopheles gambii to control malaria, and asked people to go through problem formulation exercises very much like what Monica Garcia Alonso described and other speakers described today. The third day was spent with all the various breakout groups coming back together to talk about what they discussed and to see if there was consensus of opinion on the areas that were important. We asked them to identify relevant protection goals that would need to be considered and to try and develop pathways, that should be the focus of further development. So now what I'm going to do is fly at absolutely unforgivable speed through some of the conclusions that were reported from the Reston workshop and then offer a few thoughts on how and if the conclusions were different in the African consultations. In case I get flummoxed and forget, the results were mostly the same regardless of where we did this. There were a few differences that I'll touch upon at the end. This is a publication that came out of the Reston workshop, so if you want to read this at your own leisure, you can. So there was good consensus on broad protection goals that were generalized that were relevant for this type of application. Human health and biodiversity were the number 1 and number 2, and not necessarily in that order. But universally, there was no breakout group that didn't say these were relevant protection goals for doing risk assessment for this application. Animal health, a little bit less, and partly because the mosquito has a very strong preference for biting people and only occasionally bites animals. Water quality, I won't talk about beyond this slide. It was always considered, but people had a very difficult time developing plausible pathways to harm because of the type of environment that Anopheles gambii lives in. It doesn't live in lakes and streams, it lives in puddles and garbage cans and old tires. It's a very anthropophilic species. So there were some pathways developed for water quality, and because it does have an aquatic stage in its life cycle, nobody thinks you can do a risk assessment without addressing that. But people really didn't develop sophisticated pathways to harm. So this is just a slide to tell you that there's 2 general strategies for using a gene drive to control malaria. One is population suppression. This is where the drive lowers the population of the vector and thus blocks transmission. The other is population alteration, where you transmit a trait through the population that somehow prevents the mosquito from vectoring malaria. And the important thing to know about this is that the level of exposure will be different. Population suppression is a strategy that really removes organisms from the environment. So both the gene drive as well as the existing organism are expected to reduce over time, whereas with population alteration, you would expect the population to remain stable, but you introduce a new trait, and that presents a different level of exposure for risk assessment. So human health, not surprisingly, because it was a bunch of humans in the room making these decisions, was the most frequently considered. This is a disease vector, and people are concerned about the way it vectors diseases. The relevant interaction is that this mosquito bites people, and this may sound a little ridiculous in a sophisticated audience like this, but people wanted to make it clear that that is the relevant interaction. You may occasionally inhale a mosquito, or it lands in your soup, or it flies into your eye, but under any conceivable scenario, that's not considered to be a relevant risk pathway. The mosquito is going to bite you, and that's the relevant risk pathway. Because it is a disease vector, people were very concerned and very interested in making sure that people were looking at how a gene drive would affect vectoral capacity, not just for malaria, but for other diseases that Anopheles gambii might be a vector for. Biodiversity, the two different types of gene drives present very different levels of exposure to biodiversity. But in general, experts and participants agreed that this isn't a keystone species, and I don't have time to go into all the evidence as to why that is, but almost everything we know about Anopheles gambii comes from the fact that we have been working feverishly to kill it for 100 years. So we have lots of data from places where we've had local or regional extirpation of the species for a significant amount of time, where we can look at what's happened to biodiversity in those places. And the evidence suggests that this is not the keystone of the ecology where it's found. The interactions with other species, again, that are relevant are it's either going to bite them or they're going to eat it. The other relevant interaction is that it competes in larval stages with other mosquitoes for resources, so it can be a competitive organism. It's not known to be the sole food source for anything, although there are some spiders in the Lake Victoria region that are known to prefer to feed on anophelines, but there's about 800 species of anophelines in Africa, and the gambii complex is not known to be the sole food source for anything. People don't think, again, based on experience, that this is likely to have a severe impact on any other species based on the role that this insect plays in their diets. And again, I don't have time to get into that. I will say that this is not a suggestion that people should not consider this in risk assessment. It's just a statement about what we already know. Gene flow to species within the Anopheles gambii sensulatu complex. Without boring you with the details, there's 8 species that none of us in this room could tell apart. They all look and act exactly like Anopheles gambii. And some of them are cross-compatible, so gene flow is likely to happen in the complex and unlikely outside of the complex. Horizontal gene transfer, as for almost anything that's not a bacteria, is not likely. Animal health is very similar to human health. The relevant interactions are that animals, being livestock in this case, can be bitten by mosquitoes, and people were concerned about potential for altered transmission of vector diseases. Click. Other considerations. So this goes back to some of what was being discussed this morning. I worked very hard to try to not have other considerations because I had a very defined idea of environmental risk assessment and ecological risk assessment and what that meant. But people felt very strongly that there should be some consideration of other issues. And this goes back to what is the context for decision making. So if you're thinking about using this strategy, you need to look at the risks in the context of other activities for controlling malaria. And I think one of the illustrations of that is a lot of control activities involve trying to remove this organism from the environment, and this is just one of those. Potential harms identified should be considered in the context of other vector control methods. There was a lot of consideration that I won't get into that if you try and fail to control the vector, then you may lead malaria to come back in a more aggressive way. I'm over time because I'm an awful person. Just some very quick thoughts on these activities. This is a starting point. It's very challenging to do this, and nobody thinks that we're finished with these conversations, but it is incredibly valuable to have these activities to bring people together, identify what they care about, what they're thinking about. By far, the thing that people care about most is human health and then biodiversity. Water quality was always considered, but not considered in nearly the same category as the other two things. Here's some pictures so you know that this actually happened. Thanks to everybody, and I'm sorry for going over time. Thank you very much.
Thanks very much, Andrew, and you're not an awful person after all. You're just passionate about what you do, which is what we want, right? So I wonder if there are indeed a question already with this gentleman here.
Yeah, Les Furbank from the GMO panel. Lovely presentation. It's nice to see that this is coming along. And it's also very nice to see human health listed as a protection goal. Can I ask, what are the decision-making processes if you decide to get to the point where it's okay to release them? How is that going to work in Africa?
I don't have the answer to that question, and I don't think I'm the only one who doesn't have the answer to that question. So part of the ancillary benefit of having these consultations was to get people thinking about exactly that question. And it is an open question. Some countries that are targets for this type of program have sophisticated regulatory processes in place for the use of biocontrol, for the use of genetically modified organisms, and other relevant activities. Others don't. And there is a lot of acknowledgement in the community that the intention is for these things to spread in the environment, so there's a very real possibility of transboundary movement of these things. So it's very much on people's mind. There are different ideas about how that might be accomplished, but it's very much a work in progress. And that's one of the motivating factors for starting so much before the technology is ready to facilitate these types of discussions. I think it's a very relevant thing to bring up.
Okay, if there's no more burning questions from the audience, I think we move forward. So thank you again, Andrew, very much. And then I invite Dr. Dick Shaw to join us on the stage. Dick comes from the Center of Agriculture and Biosciences International, CABI, and will present to us on problem formulation for the environmental risk assessment of weed biological control agents, insects and fungi versus weeds.
Thank you. Thank you very much. And thanks to Esther for inviting me to come along to this and talk about my favorite subject. The reason I'm talking about this is because we're doing weed biocontrol for a very long time, and it's recently become of interest to folks like EFSA and other regulators, even though we've been doing it over 100 years. So it's considered novel in Europe, and this is a picture of all the targets we've got in Europe pictorially. If some of you are botanists out there, you'll know what I'm talking about. But I'm talking about, has Mother Nature given us a potential solution for something that we screwed up? And if it does, is it safe or is it going to ruin the environment? That's the basic tenet of biocontrol. I won't go through what I'm going to talk about. I'm told to do that, but it's pointless. You'll find out. But Japanese knotweed is my particular passion, and some of the biocontrol projects I've been involved in have addressed insects against weeds, fungi against weeds as classical biocontrol agents, and fungi against weeds as bioherbicides, which is a little bit novel. And you'll understand the problem formulation as we go along. We never used to call it that. Japanese knotweed, fairly famous invasive species. It's not on the EU regulation for invasive species because it's too big a problem and no one dared put it on there, is my conclusion. But it can break through concrete and all the rest of the thing it's famous for. It pushes over walls. That's a genuine picture from Reading. I don't live in Reading anymore. And there's plenty of it on the river. I've spotted loads of it on the river. So Palmer isn't immune to knotweed's threat and curse. And as is always the case in biological control programs and invasive species, they've got lots of organisms in the native range and very few when they arrive. They've lost all their natural enemies. In Japan, against Japanese knotweed, there's 186 species of insect and 40 fungi feeding on the plant. Actually, not tourists, feeding on the plant in Japan. That's a lot. So our job is to find out whether any of those are suitable. The argument we use is they've got an unfair advantage. They've lost their natural enemies. If you have a crop with no pests on it, it grows bigger than a crop covered in pests. Everyone knows that. It's the same for invasive species, and there's all kinds of other theories behind why enemy release hypothesis is the main one that I'm interested in. The native species, European species that do attack Japanese knotweed are very few in number and don't do the job. So the question is, have some of the ones that came from the native range, could they be introduced to control the species permanently? I don't know where to point it. There we are. And our job is a process of elimination to take that 186 down to 1 or 2, because most of them are not going to be safe or specific enough to be released. And that can be done through literature review. If you find a species, you look up its name and you discover it's a pest of corn, you're not going to introduce it. You can look at field observations. So in Japan, Japanese knotweed is often growing alongside many other Polygonaceae, the plant family in which Japanese knotweed exists. And if your insect is feeding on knotweed and not feeding on any of the other 17 Polygonaceae in the square meter, it's not likely to feed on it in the real world. Therefore, it's a specialist. So that's a useful tool. Then we look at susceptibility studies. Is our knotweed tasty to the agent versus the knotweed I found it on in Japan, because there's a lot of knotweeds in Japan. And then you end up with the expensive bit, which is the quarantine work, the host specificity testing, which is basically our problem formulation. Is it specific enough to be released? are going to cut out 10 years of research with about 30 people involved to get to the end. We chose this species, Aphelara itadori. Itadori is Japanese for Japanese knotweed. It's always a good start when your agent is named after its host. So Aphelara, little sap-sucking psyllid. And then this leaf spot. The only reason you can see Japanese knotweed in this picture from Japan is because the leaf spot's on it. Every single leaf has got the leaf spot on it. So that's another exciting potential agent. And I'll concentrate on that one now. Once I've described the categories of weed biocontrol. So we have two types, inundative biological control, which is basically often taking a native pathogen, mass producing it, swamping the target agent with the natural enemy and killing it or suppressing it. I prefer to call it commercial, but I'll come to that, come to that in a minute. Classical, of course, is the mainstay of what we do, which is going back to the native range, finding the natural enemy, releasing it once, in some cases a few gravid females, and permanently controlling the organism forever. So this is the miracle, silver bullet scenario of biocontrol. So the inundative approach has a cost, and it's only ever used pretty much in high-value horticulture. There's a great example of strangler vine control in the US, where they produced a fungus that was called D-vine. They called the population D-vine, and it was incredibly successful. But the trouble was it controlled it for 7 years. So they had this massive first year, and then no one bought it again because the pest went away. So it wasn't it was a great agent, but it wasn't a good agent commercially. And it's also used where conflicts of interests could preclude biocontrol. So a good example for me is Rhododendron ponticum, rare and endangered in the Iberian Peninsula in Europe, mainland Europe, I should say, before we go to Brexit, but invasive in the UK. And that's a classic case where the conflict of interest is we work next to the Queen's azalea and rhododendron collection. So I'm unlikely to introduce a classical biocontrol agent that's going to eat the Queen's collection. So I have some sense. In my opinion, inundative control is better described as commercial control, and it's always formulated. It's in a bottle with a label. So that to me is a very clear distinction between what normally is a public good activity, which is classical biocontrol funded by governments for everyone's benefit, or inundative, which is commercial. So I'm going to go back to that leaf spot I showed you in Japan. We tested it for classical biocontrol. We weren't looking for a biocontrol mycoherbicide, but we discovered that it hit a non-target species, a rare non-target species in the UK, and we sunk it. We decided that wasn't a sensible thing to do. I'll come back to the implications of that at the end. But what we did discover was it could actually infect through mycelia, not through the spores that we were testing at the time, and that mycelia could be macerated and attack the plant. And it also had mating types. So if you only ever use one mating type, you cannot ever get the spore that can infect the non-target. Suddenly, you have a potential knotweed control agent in a bottle that cannot attack any other species except Japanese knotweed, no matter where you spray it. So pretty excited, because knotweed is a massive problem globally, Europe and North America. So we're quite excited by it. We had to rush into a patent thing because we discovered that we'd presented it 363 days ago, and we had to suddenly produce a patent application in 2 days because we'd made it public within a year. So that was quite a scary time, I can tell you. But basically the PRA is complete and it shows virtually no risk. There's almost zero risk approach to the environment. And now we have to get onto the proof of concept, get it out of quarantine. It's still under quarantine, obviously. So an interesting challenge. And we're not really set up for that in Europe, that next step. So it's going to be an interesting discussion. And if it does get considered, I would consider it a low-risk substance because it doesn't fit any of these things on the left. But if it isn't a low-risk substance and it needs to go through the full classic data package that you have with a chemical herbicide, I'm not sure what the future of the organism is because that's a big job. Anyway, same for everyone. On to the classical, Aphelara itadori. I'm sorry about the focus on this. It got corrupted, so I had to steal one from the web. So this is your invasive species at the top, and you introduce your biocontrol agent at the bottom, the blue line. It has a big outbreak population, and then it suppresses your target population below a threshold. This is the classical thing you've learned at university or school. No one ever declares what the damage threshold is. I've never seen that, but basically that's what the target is to get that permanent control that goes on forever into the car park. It's better to use a concrete example. Here's water hyacinth. Massive problem all over the world, let's not pretend. And there's this amazing weevil called Neocotina eichhornii that's controlled it all over the world. And here's the real data from Louisiana. That's 1.8 million acres of invasion. So the scale of these things can be shockingly large. and there's the control that persists to now. So that's your dream scenario of biocontrol. Is it safe? Well, it's been around for 100 years, 1,500 releases, 450 agents. 450 of those releases came from agents that came from Europe. So we've been very good at providing solutions to the problems we've created around the world, but not very good at benefiting. We've only received 4, and one of those was we released a mite against Crassula last Thursday. So this is very, very new to Europe and novel and controversial to some. We've been guided by the International Code of Conduct, ISPM 3, which has been around forever, and therefore we never use the word problem formulation, we just follow the guide. But that's exactly what we've been doing all this time, is formulating the problem and addressing it. And in all that time, there's 8 examples of non-target effects, and all of those were either predicted at the time, and so, does it eat a crop? No, release it. That was the way biocontrol happened in the old days. And they'd be predicted now. So it's a pretty solid, safe science. And the reason that is because we do this host range testing, very extreme host range testing, up to 90 species. Japanese knotweed, 92 species we tested. And we use a centrifugal phylogeny to basically, we can't test 2,000 plant species that are in Britain, so we test things that are closely related. And the insects tend to know how related things are, and they tend to be better at knowing how related they are. So simple testing, starvation test, feed on it or die. That's pretty extreme. I'll eat celery if I have to, but I would avoid it if I've got a choice. Choice tests, a bit more realistic, still in a petri dish or on cut plants or on live plants. It depends what your situation is. Overposition test, if the organism doesn't lay an egg on it, it's never going to develop on it because the egg never got there, so a pretty solid test. And then development test, if it does lay an egg on it, how far does it go? Does it produce an adult? Can the adult reproduce? So this is all basic stuff. We're not rocket science. And then for fungi, it's a little bit more complicated. We need a dew chamber, and we expose the organism in a massive inoculum load, 48 hours of permanent dew period at the perfect temperature to see if it infects the non-target plant. So if it doesn't do that in the dew chamber, it's never going to do it in the field. So very, very extreme testing. I'm not waving, it's the fly. And we applied this to the Himalayan balsam. This is Impatiens glandulifera, a big problem around Europe and North America, and we found a beautiful rust fungus that attacks the stem, causes the plant to etiolate, and then reinfects the plant. It's an ortesious macrocyclic rust, and this was released in 2015 for the control in the UK, and that's establishing another case where the rust is a better taxonomist than the taxonomist, and it detects a difference in the population with variable susceptibility. So we may have to go back to Pakistan, India, and indeed Kashmir, which should be fun, to find the right strain. The key thing is this is all done in quarantine. It's very, very hard to get realistic data from the native range. Lots of invasive species are so rare in the native range you can't find them. How could you predict they'd be invasive when you look at them in the native range and they're not hard to find? So these things are pretty hard to do. I'm on to the bit you want to hear about, the problem formulation. So the possibility, this is just direct effects. Does your biocontrol agent outbreak and attack something else that it shouldn't attack? You can test that. I've shown you the testing process we do. If it does do that, can it maintain an attack on that plant? So you can test that by seeing sequential rearing. If you stick it in a cage, does the population crash out, or does it maintain and keep growing, or maintain its level? So that's fairly easy to do in quarantine. If it does maintain attack, does it affect the population of the host plant? A little bit harder to do. You could do native range experiments to see how persistent it is, but then you've got a lot of other variables, parasitoids, hyperparasitoids, predators. You can't be spraying fungicide and insecticide all over the place. It's a little complicated, hard to do. But the question is, it's a big question in biocontrol right now, is what is the real effect? If it nibbles on something else, does it really matter if it nibbles on something else, if it doesn't affect that population of the plant? And that's another level above the risk assessments that most people do. Any non-target damage, rejected. That's pretty much what happens. So here's a lot more complicated slide. Indirect effects, apparent competition. So if you introduce your biocontrol agent, does that biocontrol agent suddenly become the prey for a generalist natural enemy that's hanging around in the environment you've introduced it into? Well, we could do that. We can find out if it's got shared taxa, whether the hosts are likely to be fed upon. So there's ways of doing that, and you can also do some experimental studies to see whether it will feed on it in choice. If it does feed on that, the generalist does feed on it, does that generalist population grow? And if it does, you can see whether it performs better on that than its normal host. It's unlikely to shift from a normal host onto the new one if it doesn't perform well on it. So that can be tested. You're getting a bit shaky now. Does it suppress the normal host? So if the you've got all these generalists, let's pretend you've got billions of generalist natural enemies because of this lovely food source that you've just given it. introduced, does that suppress its normal host? So does the phytophagous organism that it normally feeds on suddenly outburst? Impossible. Impossible to test, I would argue. You could do literature stuff, but you can't test it. You're in a lab, in a quarantine lab. You can't do population-level studies. If it does suppress the normal host, does that plant become an invasive weed because it's now got no natural enemies feeding upon it? There's a lot of ifs in this now. We're getting down to the really iffy end of the ifs. Again, you can't really test that. And then if it does that, is that a permanent effect? Again, untestable. Is the food web irreparably damaged? Something else could happen. If you introduce your biocontrol agent and the generalist natural enemy feeds on it, does it ignore its normal host and start feeding on this one exclusively? Then your normal host booms, suppresses the rare native plant, and then you end up ruining that food web. So these are apparent competition things that are incredible. There's people who work on food webs that have worked for 15 years on one food web and still don't know what's going on in the field. So you can't really do this in the lab. So it's very, very challenging. Of course, this is precluded by, the only way this can happen is if your biocontrol agent outbreaks to massive populations, it becomes a food source and doesn't affect its host because it's a specific agent. It can't feed on anything else, and its population is regulated by the population of its host. So you've got to have a population that grows massively and doesn't do anything to its target plant. That's pretty rare. So this is a very rare example, but I had to cover it because we need to look at everything. So we have a big problem with false positives from the lab. The native range studies are pretty good, but when you get into host range testing in the lab, you tend to get false positives. It will feed on something in the lab condition that it won't feed on in the field. You reveal the fundamental host range of the organism, not the realized host range, the one that will be realized after release. So that's a pretty important thing. And the realized range is much smaller than the normal host, fundamental host range. Quarantine lab studies tend to give a worst case scenario, and we tend to go with that worst case scenario, so that's worrying. And the example I'll give is rubber vine in Australia, 40,000 square kilometers of ecosystem, World Heritage ecosystem, was under threat of basic destruction by this plant from Madagascar called rubber vine. We introduced a rust fungus, and that rust fungus is now controlling it across the whole of that ecosystem. An incredible achievement. The whole size of Great Britain has been controlled effectively. And it attacked a native species in the host range testing in our lab, and it sporulated on it. And we all thought, oh, that's it. And the Australians said, we don't care. We're losing a whole ecosystem and everything attached to that ecosystem. Release it. And they released it, and it never attacked that native plant. So the testing was too extreme. So there's a danger of us rejecting things we shouldn't be rejecting. Of course, the alternative is doing nothing. So doing nothing, no natural control achieved, that's pretty much guaranteed. The invasive weed will expand, that's pretty likely. Non-target population negatively affected, of course, it's already affected and it's going to get worse. And then control costs will continue to rise. It's pretty likely unless you just give up. And then you have this serious impact. So that's just a reminder of balance. You can't sit there and do nothing. EFSA has taken an opinion and got involved in this in Trichologaster against an acacia. Very interesting project. And they recommend that the risk assessment be reviewed, not rewritten, because they're the experts. The people who wrote it are the only people who work on this species in the world. So how can you possibly produce another risk assessment? It needs to be quick because the organism's sitting in quarantine dying if you're not careful. And it needs to include benefits. The whole world looks at benefits when they look at risk assessments of weed biocontrol on public good. So an interesting challenge. It's around, it's going to stay, it's growing in Europe. The European regulation is full of species on Annex 1 that need controlling and have got potential biocontrol agents out there. So we've got to start doing it. Safety testing means we're likely to reject a good agent rather than release a bad one, so that's a reassurance, I suppose. Regulations aren't ideal, in my experience, and we need to balance risk against benefit. And I would say doing nothing is not a low-risk option with weeds and invasives. It really is the opposite. And I'll just leave you one example. You can also do the wrong thing. So here's water hyacinth. This has been blamed on the British with their second homes releasing water hyacinth into the river in Guadiana in Spain. And they had a project of pulling it out. This is environmentally sound. Look at the bank of that river. That's environmentally sound, no non-target effects, everything's great. Unfortunately, they spent 23 million doing that. 23 million euros pulling water hyacinth out of a river when there's a biocontrol agent off the shelf. And funnily enough, it came back, and they had to spend another 5 million dealing with the stuff that came back. And it's going to keep coming back. So maybe we should start looking at biocontrol a little bit more seriously. Thank you.
Thank you very much, Dick, for this very interesting talk about the weed biological control agents. And you said doing nothing is not the solution. But for the benefit assessment, who would be the best bodies to conduct such a benefit assessment?
It's very difficult because the stakeholders are so broad. Almost every sector is affected by invasive species, so you would have to have that. And the socio-economic benefits are very difficult to balance, but you can get the costs pretty seriously. There is an issue with Nagoya and ABS, because if you start putting a cost and a value on an invasion, and you control it with a biocontrol agent from another country, that country may well consider that to be a financial benefit that they want to share. It's not really, but it's an interesting challenge that we're very cautious about.
Thank you very much. Are there any questions from the audience? We have time for one or two questions. It seems not the case, and we have one from the app. Can you please? No?
Those are the scary ones.
It's none from the app.
Okay.
Okay, thank you very much. Our last speaker of today is Chris Topping. He is a professor with special responsibility in ecological modeling at the Aarhus University in Denmark. He has been working with modeling environmental impact agricultural practices affecting wildlife management for over 20 years. And his title today is Landscape Scale Population Level Environmental Risk Assessment, Current State and Challenges. And we hope to get some practical examples. Thank you.
Thank you very much for that introduction, and thank you very much for the invitation to speak. So I think I should apologize before I even start this. Following the last three talks, which were presenting extremely interesting and high-level stuff, I'm going to bring us right back to EFSA and risk assessment at a very simple level, I'm afraid. So I want to talk to you about the landscape scale and population level risk assessments. Tell you a little bit about where we are in a very EFSA-centric process. So I'm not going to give you the big review across the world, I'm afraid. So you're probably all aware, and you've heard a lot over the last day or so, the focus in environmental risk assessment is changing. We started with this very traditional laboratory approach. We make some generalizations about fate and exposure, and we come to a risk assessment. And more we're being inclusive of other things, particularly ecology, detailed fate, detailed exposure modeling. And now we're talking about population level, landscape scale risk assessments. And as you've heard over the last couple of days, we're now really also talking about introducing social science. So I'm not going to go there. I'm going to stop at this point. But you can see things are really changing. And this is being termed in the last couple of EFSA outputs as a systems approach. So this views the receiving environment in a very different way to the traditional approach. We're talking about landscapes. We're including multiple stresses, not just regulated ones either. We have a longer temporal perspective than we normally use. It's a spatial assessment, so it matters where are the organisms, not just how many there are. And it includes the connections between the parts of the system. So it includes the feedbacks. And I remember there was a question this morning about how we might do that. This is an integral part of this approach. Now, as Jörg mentioned, the traditional and current risk assessment for pesticides is rather straitjacketed by the guidance that we have. So basically, if we start to take the systems approach, we're going to have a fundamental change in the way we set up and carry out the environmental risk assessment. So, but this is a fundamental change based on what we have right now. It's perhaps less advanced than the things that we've been talking about over the last couple of days. So we're somewhere in the middle. Okay, thank you. So this is all I'm going to say about the current capability. I'm going to just make the bold statement. We have the tools to do this. We can actually make these fully integrated landscape scale risk assessments at population level including very highly detailed dynamic landscape models. So this is these models which are perhaps at a 1 meter resolution over a very large scale. It might be 10 by 10 kilometers or even up to 50 by 50 kilometers. They're real areas of the world. These are not abstract models. And we combine those with agent-based models of organisms, which allows us to make very detailed predictions about the way organisms are going to behave in that environment. And because they're real areas, we can test them. So this we can already do. And we have tools already for a number of focal species. So we're already more or less ready to use. We've got skylark, we've got hare, field vole, and a beetle. And we have a number of other models which are sort of waiting for the final development, but are already existing in one form or another. And there's actually another whole pile of models which are being started with new projects. So we have quite a range of focal species tools already either developed or underway. When we come to the dynamic landscape modeling, we've got models now with national coverage in Denmark, in Poland, and in the Netherlands. And it's underway for Portugal. And again, new projects starting to add another 5 EU countries. So basically all of those green areas on the map there are countries where we should, over the next few years, have national coverage with these very detailed models. So we're doing pretty well. But obviously when we start down this new approach, we have new challenges and we have some opportunities, of course, which is why we're doing it. So I'll take the opportunities first. These are much more ecologically realistic assessments. So these are these kind of mechanistic models that Valerie was asking for this morning. We have the longer term perspective. We've also got the options to tailor the risk assessment procedure to local conditions, and we have the potential to include mitigation inside the same tool. So there's some extra utility involved in using this approach. But exactly the advantages of this bring some problems when it comes to defining the risk assessment, because suddenly everything is open to question again. We're really changing the basis of the risk assessment. So we have a conceptual problem in actually defining environmental risk assessment. We have obviously issues of people accepting and using the new tools. And then slightly more boring, we have the technical issues required to develop and find the data, and logistical issues. Basically, we need to get resources to do this. So I'm just going to mention the bold ones very shortly, and I'm not going into details on any of these because there isn't that much time, and they're big subjects. We're looking at the opportunities. So ecological realism basically means that we're including the interactions between the components and the context of the environmental risk assessment. This is the most important slide that I'm going to show. So in this little graph, you can see we've run some scenarios. We've run 2 scenarios for new populations, and they're the colored bars. And we've done it for 10 different landscapes in Denmark. Now, this is the same scenario, and the important thing is to see that the colored bars are different lengths. These show the impact on the new populations. Longer bar, bigger impact. So the landscape context structure matters for the outcome of the impact assessment. But most of them are correlated. So most of those blue bars are correlated with the orange bars, except for the circled ones, which are not. So there's also interactions. So this is part of the problem. So we have landscape composition not only altering the impact, but also interacting with the scenarios. I mentioned the long-term perspective. Here's one of those scenarios again. And what we've done is we've run the scenario at year 11. We've started to spray pesticide, and we've either done it for 1 year, 10 years, or 20 years. What you should see from this is that, firstly, when we start to spray the pesticide and keep on doing it, impact increases. So our current risk assessment looks at a single application, basically, a single year. But if we keep on doing it, impact will often increase. And the other thing is that the recovery takes a long time. And this is quite common. And these patterns that you see here are common for many species when we look at these. So I mentioned we have an extra utility. So we just saw that landscapes and agricultural systems alter the predicted impact. So that actually means we can use that. We can use that fact to tailor the risk assessment to specific requirements, say, of member states. So if we've got any systems or locations of particular interest, we can actually model those and do the risk assessment exactly there. So we can make it much more relevant than it is currently. And so you see an example here of how different landscapes can be. These two are both from Poland. They're both the same size, but the structure of these two landscapes is incredibly different. Another utility part, I said we can include the mitigation in the same tool. So we would normally be at a situation like this. When we run the risk assessment, we might be interested to know how does the population change in size, but also where is it? Where is it changing? Are we reducing the distribution of the population? So we get a graph like this. And without the pesticide, we would be at the origin, obviously no change. If we apply the pesticide and get something like this, we might find that we've crossed any threshold we've defined and gone into a situation where we would fail the risk assessment. But we can just say, okay, let's put some mitigation into the model. Let's put a field boundary, a sown field boundary around all the fields where we spray the pesticide. What effect does that have? Makes it better, but it's not good enough, so let's make them wider. So what this means is that a future system might be taking this kind of information into account, and you might get a label restriction which says, you can use this pesticide, but in this region you have to put a 5-meter field margin around the field if you're going to do it, or something along those lines. So challenge is, the worst one is this conceptual challenge, I think. We have to redefine a lot of things. So here's just one example. Protection goal definition. If we want to define a protection goal at population level, it might be population size, but how much? And is it a relative effect or is it an absolute effect we're interested in? So here's a little example showing how this might be important. This was field voles in orchards, and we sprayed in the simulation with an endocrine disruptor. And then we manipulated it a bit. So we took the optimal habitat for the field vole and we either moved it close to the orchards or we moved it away from the orchards. And the random was in fact the real case. And we got 3 different impacts. So here we start spraying the pesticide in year 5 and the population starts to decrease. But it decreases most where we have the population the habitat moved away from the orchards. So, okay, that's where we get the biggest effect. Well, not really, because the actual net effect, the number of individuals that are affected, was largest in the scenario where we move the optimal habitat close to the orchards, because that increased the overall population, so more were exposed and more were affected. So we need to decide what is it we actually want to specify as the protection goal here. We get different answers depending on what we choose. But probably the vole isn't the right species to use. If we want a population level effect, do we want to use an R-selected, highly fecund, highly dispersive species, or one that's perhaps more fragile? So we need to consider that. And then we need to consider everything else about what to include in the scenarios. And I won't go into that, but you can imagine there's a lot, especially when we start to talk about multiple stresses. But if we can do this, then we actually have another opportunity, which was also mentioned, I think, this morning. And that is that if we can manage to set these 3 things, we can use the simulation to set laboratory toxicity thresholds. So we can actually use this to drive the risk assessment the other way. So basically we can simulate taking the environment into account, and then we can set the laboratory toxicity threshold by finding the toxicity in the simulation, which gives us the maximum impact that we're going to allow. So that might look something like this. This is an example for eggshell cracking. If we take a realistic worst-case scenario and for landscape selection pesticide, and we take 2 assumptions about distributions of effect because we're not sure, and we make just this is just for illustration only, an assumption that we'll allow a 20% population effect, then we can simply read off the allowable amount of eggshell cracking that results in an impact of 20%. And that gives us a threshold for toxicity testing. So this would be an example of how you might go about it. I don't suggest we have a 20% impact on skylarks, by the way. This is really only for graphical purposes. So we have some other opportunities as well. The biggest, the big challenges is acceptance. And actually, 4 of the recent EFSA outputs have recommended this kind of landscape scale modeling approach. But we don't have tools and we don't have guidance. So we have an EFSA implementation strategy, which is an EFSA PPR panel activity. And the aim there is to provide standard tools for environmental risk assessment at this scale. And the strategy is basically to use the things we've got and build on those. So we're not starting from scratch and reinventing everything, but we're taking the building blocks we already have. And the idea then is that we can provide risk managers in Europe and member states with the tools for landscape scale risk assessment for active substance approval and authorizations of pesticides, and also develop landscape contexts that could be used for higher tier risk assessments. So this is what we want to try to provide. And basically to make this happen, and this is the last slide, basically we need to fill in a bit more of the green. It will be very nice to get full coverage in Europe. We need more species. You'll notice I never mentioned aquatic systems. This is because I don't have any aquatic systems at the moment that are integrated in this approach. We need those. This is a priority. But the most important thing is we need active participation of the member states and other potential users in this process in defining how we're going to use these tools. And I think this is probably one of the biggest challenges in actually figuring out how we're going to do that. So our strategy is to make the tool available and then open a discussion and hope that will lead to an accelerated take-up and lead us towards actually getting these things used in practice. So thank you very much.
Thank you very much, Christopher. So Christopher is offering us the opportunity to lift our environmental risk assessments up to the landscape level and the population level. Does anyone have any questions about that? There's a gentleman in the middle here.
Thank you for a very interesting talk. I'm Leibert Brussat from Wageningen University and Research. Now, this morning we also considered the landscape level, but then in terms of ecosystem services. And you do a similar thing, but then focusing on an array of species. Mm-hmm. So could you reflect on the utility of these both approaches?
Yeah, so we're approaching, I think, the same goal from different starting points. Sorry, is it closer? Oh, we're approaching the same goal, I think, from different starting points. The starting point I have is the current risk assessment scheme where we are focused on focal species, and I'm trying to build this up to increase the range of species that we have in order to encompass hopefully the range of focal species which might be relevant for ecosystem services. So actually I didn't mention, but a lot of the new species we're starting to work with are non-target arthropods, beneficial arthropods, so predator species. Directly of pest species. So I guess we're nibbling at the problem from both ends, but neither approach is going to cover everything at the moment. So I think that I see these things as being complementary, and eventually I hope they'll join.
Thank you. There's also a question here from the gentleman in the blue shirt.
Thanks, Chris. It's Peter Dom, BASF. Chris, you have used this model for a number of species, and in some cases you also have really shown very nice correlations between your model predictions and what you see actually in your tested environments. So you tested the model. Here you showed again the nude model, and I've seen that people used your model to say there is a proof of the effect on the nudes. So has this been tested already in Denmark, in the region where you have all the nudes?
No, so I used the nude model because I wanted a graph that had that nice bar distribution. I could also have used the hair, but no, the nude model is in the second category, so I had it listed under the ones that are still under development. So it's quite well developed, but it's nowhere near ready for use. So none of the graphs or numbers that I gave are useful for anything at all in their own right. They're all illustrations of the kinds of effects that you can generate. But I'm really careful at the moment. This is not something which we're using in risk assessment. We haven't decided how to use it. So I'm doing everything in a very abstract way.
Are there other questions from the floor? I actually have a question myself, if I'm allowed to take my prerogative, which is, Christopher, so these are simulations. So my question is, in order to feed into the simulation, the parameters, you must have a very sophisticated understanding of the ecosystem dynamics. So what do you base that structure of the simulation on?
Yes, so there are two parts that we work on. The one is the landscape, and that's a very, very detailed simulation, including the farmers going out every day and doing everything they would do on the field. It simulates the crops growing on every field in the landscape independently. So we have an environment which we can then spray the pesticide in. We even have soil types, and we can follow the pesticide fate in that environment. So this is the one part. You need that to drive the simulations at all. Mm. The second part is the animal models, and these are these mechanistic animal models, but these are very, very, very detailed. So we're simulating, again, daily behavior of the animals, where they move around, where they feed, how much they feed, where do they breed, when do they die. And this baseline simulation, we combine the two of them, is what we can test in the real world. So we can go out and we can say that, yes, these are behaving just like the real world. If we have that level of confidence, then we have a good starting point to start the risk assessment and say, now if we change that situation, the model is likely to respond in the same way as the real world would. And then, ideally, after that, we could monitor and find out if it did. Mm. This would be the ideal solution, and we would end up in this iterative cycle that Valerie mentioned this morning.
There's another question here from the...
Yeah, this is, Tobias Frische, German Environment Agency. I have the following question. I would like to know if you proceed with this, what you're proposing and try to model, so to say, the most realistic situation in a given crop. Do you have the data in the end to do it? For example, for a crop like an orchard or a vine, where you have a spray series of XYZ number of pesticides coming in. So you need the application data, which must be, so to say, a kind of realistic worst case or within the spectrum, and you need the tox data and so on. My question is, so if you want to go this way, do you have the data?
Do we have the data? Well, we have some data, I should say. The way that what we don't have, and we would very much like to have, would be the data that which is collected under the Sustainability Directive, which is not collected in any standard form and isn't, as far as I can see, used for anything anywhere. But this would give us the actual amount of pesticide and the types of pesticide that are sprayed by all the farmers. It is recorded, it's just not used. Nobody has it. The next best thing, and the thing that we use, is that we go to regional advisors, agricultural advisors, and we find out from them what are the farmers actually doing, and we create basically probability distributions of applications for different products, and then we combine them. But when we come to the toxicity, then we have another new problem, because what do we assume about the toxicity when we make the baseline? Well, at the moment we assume that all the things that are out there already and have already been authorized have no toxicity if they're not supposed to, or have a minimal toxicity if they're allowed to have that. And this is one of the many things I said we have to really rethink where we start, because I have no idea really how to get the real toxicity data into the simulation. In the same level of detail as I can do for, say, the animal behavior. So this is a challenge.
Great. I think we have to close the discussion there. Thank you very much, Christopher. And we now move on to our panel discussion, and I hand over to Dr. Wendy Craig from the International Center for Genetic Engineering and Biotechnology.
I want to thank you. Oh, well. Thank you, and stop. We're short of time, and I want to thank you for not stumbling over the name of my centre, because I stumble over it as well. I'm also mindful that under instructions, the session has been planned in order to inform EFSA of current challenges and future developments in ERA. So I'm going to try and keep the conversation focused on that. But of course, at any point that it can be open up to the audience, I'm welcome to read from the app or take questions from the floor. But because of the time limitations, we're running a little bit over. I'd like to have the discussion at least touch upon 3 underlying themes that I recognise. One is the identification of the environmental risk assessment needs for future products. Is ERA already fit for purpose, or can it become fit for purpose for these particular products? I'd also like the discussions to reflect upon the robustness of problem formulation. Can it be used, for example, in other regulated technology sectors? Can we learn from one another in that regard? And is it a good way to identify data for regulatory purposes? And the third one that I'd like, hopefully we have time to cover, is what is the impact on problem formulation in the absence of clear policy goals and decision-making criteria? So they seem to be the 3 main underlying themes that I picked up on. There might be more, and I'm open to have the panel discuss them as well. So I'd like to invite the panel up now to the stage. So can we have Monica and Jorg back? We met them this afternoon. And also Phil McDonald, who was introduced yesterday from the CFIA. If you can come up, please. So I'm going to pick on Phil because you were the last one up to the stage, if that's okay. In that, just for clarity purposes, because I'm aware that in this afternoon session, the focus was problem formulation, and many of the presentations had problem formulation in their title, but not too many of them explicitly stated how problem formulation fit in with what they presented. So I'm just wondering if you could just take just a couple of moments just to give us an understanding of problem formulation from a regulatory perspective. Why are everyone interested in it? And for example, what use does it really serve to regulation?
Thanks for picking on me, Wendy. So I think actually I was grateful that my colleague from Canada gave I thought really kind of an elegant demonstration of a risk hypothesis and a pathway to harm. And I think what that illustrated is one of the powers of problem formulation is to take difficult issues and break them down into manageable chunks. And I think what Monica said is also, it's a useful message, is that risk assessors don't explicitly always use problem formulation, but implicitly always do. But what's really useful about that kind of, that really structured form of problem formulation is it's very good to be able to communicate to risk managers and to decision makers exactly what the thought process was. And I think the nice thing about that example was, first of all, is you can't invalidate the risk, the pathway to harm, so that's useful. And second of all, there's a risk management recommendation that went out of there, plus a way of communicating uncertainty. So I think when you capture those things together and what the policy objectives are and how your policy objectives, your protection goals have actually guided problem formulation, it makes it a lot easier to communicate really fairly complicated things, and especially something like transgenic salmon, for example, where you've got very politically charged overtones to that. You can make a really useful recommendation to a decision maker.
So just to keep the conversation moving on, we were presented with a number of quite diverse case studies, especially in this afternoon session. And so I'm wondering, Monica, if I could turn to you. Based on your experience with problem formulation, how do you perceive that problem formulation can actually contribute to improving environmental risk assessment and decision-making across different technological sectors that are regulated, if at all possible?
I think that the main advantage probably is organizing the risk assessment in a logical way, so making it more transparent, so you can clearly determine why, what you assess, why you assess that, and how you went about the assessment. So it makes it more understandable from both sides. So it does improve its purpose, which is the facilitating the decision making. So when you have discussions with the regulators, they can actually pick up on your logic and why did you choose these protection goals, why did you choose this particular hypothesis, and if you have thought around that, then you are more likely to have done a risk assessment that is actually going to satisfy them, and they will feel more competent or more supported in terms of making their decision or recommending a decision to the risk managers, depending on how the regulatory system works.
So is this, my understanding therefore is there, that in other technologically regulated sectors, it's not that they don't use problem formulation, it might just be that it's not so explicit. And that, I especially liked your presentation, that it really laid out a kind of map as to how you would organise the approach to the risk assessment. Is that something that, did I just perceive that myself, or did the speakers in the session also agree with that, or are you do you have a different position to that? Okay, because I don't want to dominate, it is a discussion here. So I will come out to the audience at different times just to ask for your input. What I'd like also to do is that I picked up on a question, especially during Jorg's presentation, and it was more to do with identifying what in Monica's presentation was called operational protection goals, and how they are a type of translation from a more overarching protection goal that maybe the legal frame gives. So can you give some insight or some reflection on how those operational protection goals are identified, and is that just something that happens within a dialogue between risk assessors and risk managers, or is there a broader consultation to understand what would be the most best fit operational protection goal?
Yeah, okay, I think I have to start a little bit earlier even,
okay,
because I made the statement that I would like to see the same protection goals applied across all regulated entities and even beyond, because we have unregulated entities that do harm and nobody looks at them because nobody's asked to look at them. So from a nature perspective, I want bees to be protected from whatever could be harmful. But this is an ideal world. Huh? And what I forgot to answer to this question that was addressed to me earlier is that sometimes the policy protection goals are already different. You know, so the fact that we are targeting different or addressing different protection goals in a GM unit at EFSA and in a pesticide unit is sometimes due to the fact that the policy protection goals are different. So we have to start much earlier and to rethink on this level. Also, the risk managers that EFSA, for example, is reporting back to in Brussels are different when we talk about pesticides and GMOs. But we are still always talking about the same environment, and I find this very worrying. And in particular, an example from not my research, but research in my institute that I like to mention here is some years ago in Switzerland has introduced new mowing machines that farmers could use to have their compensation areas, you know, mowed like twice a year. And they were so fast that bees, honeybees, couldn't escape anymore. And a colleague in my institute has done a study that if a farmer does the mowing on a sunny day, what they like to do, you know, they killed on average 10,000 bees a hectare. And I got upset by that because that was the time where we were really worried about Bt maize effects on honeybees, that there might be something. You know, they were killing 10,000 bees a hectare. And I was, you know, confronting regulators with that, and I was really shocked. They said, no, but we don't look at it because nobody asked us to look at it. But that's absolutely understandable, I learned, from a regulator point of view. But this is the situation we are in. I think this is very unfortunate. So we should start at that level and see that we are kind of concerned about the same things independent from the technology. And then we are still struggling to get back and see how can we make this operational, and how can we formulate specific protection goals.
So is this something that you feel is a Eurocentric problem, or is this something that regulators in other parts of the world are struggling with? Phil, if you can reflect on this.
So I think Jörg hit really very much on what the fundamental problem is here, and that we have different legislative frameworks. We operate with very different structures and regulations, and some of the regulations are very, very old, and some of them are much newer. So SEP is a good example, and sometimes we've tried to retrofit things that we already do. So I think, you know, your perfect world is a good example, and I think the discussion certainly heads us there, and I think we try to work more and more towards harmonized thinking, and I've heard some really interesting thoughts here today. So I think if my take-home message maybe from this meeting would be that it's not that we're starting from nowhere. We have protection goals. We do use problem formulation. We do environmental risk assessments, but we can certainly do things better. So I think some of the messages that you had about trying to incorporate things about ecosystem services, some of the things I heard, when those become more generic messages, it's going to be easier to think about maybe how we do ERAs in a more systemized or maybe a more logical manner. But I think just to finish off, I mean, I remember back in a discussion with someone who conducted the farm scale trials for the UK, and we were talking about, you know, the impacts of GM on farmland birds, and I said, well, did anybody ever go back and look what happened when you started adopting widespread winter cropping in the UK? I bet that had a huge impact on farmland birds as well. So, you know, our perspectives and our focus is also different when you start off the risk assessments.
And so it's a general question to the panel. Do you each perceive problem formulation as a useful tool in being able to at least provide a good focus and maybe strategic approach to the risk assessment that's maybe geographically independent? It can cross different environments, different nationalities, regardless.
So I find problem formulation like the going back to basics. So when you learn a risk assessment, you learn how to do risk assessments by basically learning about problem formulation. So I think is instead of trying to make fit a system that you already have for risk assessment for a particular stressor to another stressor, you take a step back, you go back to problem formulation, and then you look from that perspective to see what you can do in terms of assessing the risk for that new stressor without reinventing the wheel, but also applying the real basics of what risk assessment should be about. So I think that that's the advantage, and that's why it's becoming quite popular for products like genetically modified crops, but also, you know, for other products that are coming through, because it's just going to this back to basics of risk assessment and looking at how you can actually facilitate this decision-making process.
There is a question from the app at the moment from Anna, if that can be shown on the screen. And Anna's asking to all of you, what kind of simple tools you might suggest to use in developing countries, for example, to be able to predict risk. And the example here is of chemical exposure, but now that we're trying to broaden problem formulation and environmental risk assessment across different regulatory sectors, is this one of many tools, or is it the principal tool, or is it not fit for use in developing countries? Or I'm just trying to open up the question a little bit more here.
Well, I would say that problem formulation would be a perfectly good tool. I'm not an expert on chemical risk assessment. You know, I know a little bit of pesticides and a bit more on genetically modified crops. But again, I think it's just going back to basics, to looking at how you're going to evaluate exposure and hazard to then evaluate the risk.
I'd certainly agree with that. So I think, I think when we started looking at GM crops, it was kind of evolved in isolation. And I think Monica's right that people went back and looked and said, well, what we're doing now is not really working very well. So we need to maybe take a step back and figure out why it's not working very well. So I would say the same thing. There's when you're looking at something like really chemical exposure, and you have a complex and a larger problem, the beauty of problem formulation is exactly that. You can start thinking about that in simpler terms and break something which is larger into the little more digestible chunks, and then try and figure out what are the things that you really care about and how are they likely to be impacted and how are they likely to be impacted. So I think problem formulation is a really good way of getting it, and I'm not a chemical expert either, but I think it's a really good way of getting at things like that.
And I can maybe just add, I have been involved in several workshops in developing countries organized by ILSI, by the Research Foundation that Andrew represented in the past, and I found that I was always surprised it was on GMOs, but not on pesticides, but that doesn't, I think it similarly applies. I think the nice thing there was that you had a lot of people that have a lot of concern, and they could put the concern up on the table. You were building these pathways to harm, because you can do that. You know, this is a very simple exercise. And then try to find out, are they at all plausible? And you will be surprised, a lot of concerns were built on pathways to harm that without collecting any data where, you know, people got that idea that they were actually basically nonsense or very, very unlikely. And so you can use this exercise within a few days to figure out what are the real things that should be of concern, where we should collect additional data or go after. So you can do that with a lot of people that are relatively, let's say, ignorant, you know, don't have the details, but you can use this as a tool to solve a lot of problems and address a lot of concerns.
Okay. We have another question from the app, and it's from Peter. And Peter's saying EFSA has invited a number of presentations on GMOs, which he believes are very interesting and good talks. But why the emphasis on GMOs, who he says that are taking the effort to trying to register GMOs in the EU? Do you believe that the newness of the regulations concerning GMOs has allowed the introduction of newer approaches to environmental risk assessment, that the other technologies whose regulations are maybe more long-lived can learn from that and adopt, or is it something intrinsic about GMOs that needs this particular approach?
So maybe my experience is, and I think you might be right with that, my experience is, or I got in contact with problem formulation in the context of GMOs, which is a kind of a new discipline. And when I have been in discussions within EFSA with other units, you know, even pesticide people told us, you are in a good situation, we are stuck with this old regulation and the data requirements, but you have this fortunate situation, you can start thinking about the problems and protection goals and so on. But what they forgot is that they have done problem formulation and thought about protection goals long time ago, you know, so they are testing these two species that I showed in a tier 1 study because they are concerned about biological control, which is a protection goal, and you know, and but they are not explicit about it. So I think this problem formulation, as we talked about it, is used very explicitly in the GM world, but it has been done without really referring to it in many other disciplines. And I think that the nice thing now is if we put it on the table like you did for CABI, you know, I think it helps us to discuss among each other. So it's a communication tool and to progress together.
Just to quickly add, I think that the why is, as Jörg says, you know, I was an environmental risk assessment for pesticides for 10 years, and we never gave a second thought to problem formulation. We just got on with it and did it, because we had like a set of data requirements to fulfill, we had our trigger values, we had our criteria to go ahead with the risk assessment, and it wasn't until I went into the risk assessment of GMOs that we encountered this need to actually communicate and organize and make the risk assessment more fit for purpose, because it wasn't at the time, and that was 20 years ago, and we're still talking about problem formulation. So I think that, you know, in asking us, I guess, to join this was to share this kind of information with you of what we went through in terms of finding our way through problem formulation, because now we have other emerging technologies that might not have a fixed way of doing the risk assessment, but could benefit of these tools that have been developed to then find their way to fit for purpose risk assessments for those types of stressors.
So I'm going to take the advantage of putting a very Canadian spin on this, in that for us, we were presented with a very interesting challenge in that we don't regulate GMOs in Canada. We regulate a whole range of plants with novel traits, and then on top of that, we're looking to figure out policies on how to deal with new crops. So things that came in that people wanted to adopt whole new types of crops, and then some of those new crops were also going to be GM. There'll be platforms for producing new kinds of products. So we were trying to look at techniques to try and harmonize the pest risk assessment world and the GMO world or PNT world together, and we landed right firmly on problem formulation as being the place where you could take sort of differing world views, trying to keep something out of the country and trying to figure out whether it's safe to grow something there, take those kind of differing world views and meld them together into something that made sense.
For those of you who know me well, I can talk about this topic for forever, especially when there's beer or wine involved. But I've been reminded that unfortunately we just had a short amount of time available for this discussion, and I have to bring it to an end here. But I know that we'll all be here for the next few days, so I'm sure that you'll be approached over the coffee breaks, lunch breaks, etc. So I'd just like to draw the discussion to a close and to thank you for being so open to coming up and airing your views and opinions on it from the front. Okay, thank you. I pass over the floor to Catherine now. Yeah.
Thank you very much. I now have a few minutes of your time before the drink to do the wrap-up, which I'm going to do rather quickly because we are a little out of time. Thank you very much to the audience and all the speakers for your participation. I think that we have consensus amongst us on the value of the problem formulation, developed for pesticides, but now transferred to a number of new technologies. It provides us with a logical frame for organizing our knowledge and for framing the assessment. It allows us to produce environmental risk assessments that are reliable, consistent, and fit for purpose, and that can be transparently communicated. So I think we see a momentum towards harmonizing our use of this method across different risk assessments. I want to just raise 3 particular points. My first point is about protection goals. We repeatedly returned to this need for clearly defined specific protection goals that can be transferred into operational goals that can then be turned into assessment endpoints that are measurable. I think this was a point that came across in a number of the presentations. And that we can take a consistent approach using the problem formulation, assessing across these different endpoints. This allows us to then develop these pathways to harm. And I think we had some really nice examples of that this afternoon with the different presentations. And that these pathways to harm allow us to map out the kinds of knowledge that we will need in the risk assessment, and to think about the possible tests we'll need and whether we need for simulations. My second point then teases out a few of the challenges that I saw. So I think we had a number of frustration expressed about the lack of clearly defined specific protection goals, ambiguous endpoints that we can't measure. We also had this point about protection goals being different across different technologies, and this being rather unfair. It creates an unfair playing field for different emerging technologies if the protection goals vary. And then we had some more specific points about, are we using the right tests when we pursue the testing against these different endpoints? What's the role of precaution? We had this point about assessing the benefits. My third point, my last point, looks at the dynamic between the risk assessor and the risk manager, because I think this was really at the crux of some of the challenge. And we repeatedly heard that the risk managers have to set these clear protection goals. But it seems to me as well, listening, that the risk managers also have to have adaptable protection goals that can adapt and respond to emerging new evidence of the complexity of ecosystems, better understanding of the risks. So it seems to me that it's a dynamic relationship, and that what we really need is effective collaboration between the risk managers and the risk assessors. And that dynamic doesn't happen in isolation. It sits within science outside regulatory science, exploratory science, which might come up with new understanding of risk, new perceptions, and that could then feed into the risk assessment and indeed the protection goals. But it's also influenced by the social context. And I think this issue of values that we really talked about a lot yesterday comes back into this dynamic relationship between the risk managers and the risk assessors. Why is it that we are more conservative, more precautionary about certain technologies over others? If we were to do a benefit assessment, how do we manage those trade-offs? That should be in the risk management domain. But how do we then manage those values creeping into the risk assessment? What's the role of precaution? What false positives are we willing to accept? You mentioned this example with the weeds. So I think my final point is that I really see a need here for collaboration between the risk assessors and the risk managers in defining these protection goals and the level of harm that is unacceptable and acceptable. That was everything I wanted to say in summary. I have some housekeeping messages for you, which are that now we can move to attend the poster highlight pitches, which will go until 6 o'clock, and that after that, we have shuttle buses to ferry us into Palma for some delicacies and wine, let's hope. And that tomorrow we resume at 8:30 in the morning, bright and early. So it just remains for me to thank our colleagues at EFSA. Jan is here. He was very instrumental in arranging this session, so thank you very much. And thanks to the audience and the speakers.